Hello, everybody. I hope it's not too disappointing that I will talk in English. My Russian is not so good. And I want to thank the Traktoria Foundation and Konstantin for inviting me and making all this possible. So I will try to, so it's the first time I give this talk, so it's also the first time for you to listen to this. And I will put a lot of effort in trying to be easy, and if you didn't understand something, ask me a question later. And the topic of the talk is that you have the question about how to make an animal, right? It's here symbolized with a familiar, you're likely more familiar with the egg than the chicken. Depends where you grew up. And and this is the major question, and I want to give you an insight into how we nowadays understand how an animal develops. And one of the interesting questions is, of course, also how do you make this diversity, and how do you make different bodies of animals in the development, and how are these patterns generated? How do you make an animal which have 6 legs, and or animals which have 4 legs, or even wings, and things like this? So here you see a broad diversity of how animals can look like, and there is, of course, a much broader diversity as well in nature. We have about 10 million species estimated of animals, and but we mainly study only a few of these animals, which is mainly the fruit fly you see here. The fly you can detect, right? Because I don't have a pointer here, maybe I will use one, but it's of course difficult with these backlight screens. And the fruit fly is well studied, and here you can see a rabbit as well. So people usually study the mouse or rat development instead, but there's only a little bit of embryology known of rabbits. So the questions is, of course, so humans are also animals, and you see, this is the way how you make an animal, and how you were also made over some times of like 9 months, and then you hatch, kind of, and then you grow up, and you are able even to listen and understand these talks. And I try to pin it down to the main question, which is like you need a transfer of information to make an animal. So from an egg to an adult, you need information, okay? And this information has to be transferred as well. So you also need communication. And then, of course, the main question is, so what is this information which, for example, makes the position of the arm? How is it during embryonic development made clear, and also safely clear, that the arm is growing here and not somewhere on the back? Then, who is communicating? It's like, what are the units of life, and how do you organize an embryo like this? And how is this information communicated? So what are the mechanisms used to position and transfer the information between the units? So, and it should be clear, and this is not only my bias as a molecular biologist, but the information is carried by molecules, okay? So here, I show you different kinds of molecules. In the middle, you see the DNA, the molecule, the huge molecule you have packed in chromosomes in cells, and this is kind of the backbone of the information, the storage of information, right? So in this DNA molecule, everything of information is stored, and then it is recalled during the development, and also recalled now in your cells, because this is also important. And then you have molecules, which up there is hormone. And hormones are also essential molecules, which also make it clear how, for example, a caterpillar turns into a butterfly and when this timing happens, right? And this is exactly the ecdysone, the molting molecule, molting hormone, which insects are using for making and triggering the transformation from the caterpillar to a pupa and from the pupa to the butterfly. But these hormones have to be also received, so the information has to be received. And this is down there on the bottom right. You have the ecdysone receptor molecule. So this is a huge protein which then receives this signal and then tells the cell what to do after. And then we have also smaller peptides like this one there, which is a neuropeptide, which is used for signaling between the nervous system, for example, and other areas. So different kinds of molecules are important to pass over the information between the units. And how the storage is transferred is that you have this huge information in the chromosome, which is here, the DNA, and then this DNA gets transcribed into smaller molecules, which is this mRNA or messenger RNA. Messenger is clear, so it's transferring the information from the nucleus to the areas where the proteins are made. So when you eat protein-rich food, this is mainly the stuff, what here is the protein. And the proteins are the machines, right? They are the executioners which make then specific processes and also build the information as well. So now I show you the key players of development, and these are cells. So the cell is the unit of life, and it is in principle everything what is not a cell is not a living being, okay? But everything what is alive is also a cell or contains cells. And when you stain them with some colors, they look beautiful, of course, but there's a lot of more information in these cells. And here I show you a cell, which is a cell, kind of an adult cell, which is in the body. And here are some features, what they can do. So the storage is in the nucleus, this pinkish area. And then you have the membrane outside. And this membrane has also holes and some receptors, which can bind where these other signaling pathways can bind. And they also can secrete these cells, so they can release other molecules. Okay? And this is the key play also in the development to make an animal, because an animal is composed out of many, many cells. And now I show you, and you can guess, right? Because I said not all animals or things are always composed out of many cells. But sometimes you have here a single cell which is predating on a multicellular animal. So this poor thing contains about 600 cells, which is a rotifer, a wheel-bearing animal. And the predator is only a single cell. So this is a ciliate, which is, you can see also the size. So this is about the same size, but one organism is only one cell, and the other organism is 600 cells. So one cell eats 600. And when you wait a little bit, then you will see that this whole organism can swallow, or the cell swallows the other animal and can eat it and digest it. Yes. No, not vice versa. So the single cell on animal, which is the one on the right, is eating the other animal which has 600 cells. Okay? So this is to show you that a single cell can be very, very complex and can do many things. These are highly specialized, and here it swallows it. And these are highly specialized cells sometimes, but during the development and how to make a multicellular animal, you start from a simple organized cell which has many functions and can become many different things. So what is necessary then to make a multicellular animal, right? Because when an animal can also be only one cell, so what is important that you can make many cells, right? And the essential part is that cells, to build us, for example, have to cooperate and make also shape, right? So although this single cell you have seen in the video before had already a specific shape, but to build our body, it takes much more communication, information, and shape building. So cell-cell communication is essential for making an animal. And now I show you a short animation, which I made by myself. So this is a cell talk, right? You can see it. Cell talk, which means the left cell, which is symbolized by the square, will talk to the other cell, to the right cell. And they do this using molecules. And I mentioned that the molecules are the carrier of the information and that there are different molecules in place. So now watch what happens with the signaling factors. So these signaling factors bind to receptors. These give a trigger to the cell inside, and then there are transcription factors binding to the DNA and making the cell different. So you saw the change of the color, which indicates that the neighbor cell gets the information and does something and becomes maybe a different cell. And this cell-cell communication is happening. So the key information here on the slide, although it's a little bit complicated, means that it's always the same mechanism. So you start with a ligand, which is a signaling factor, which is floating around between the cells. These ligands bind to receptors and to the cell surface. And not all ligands can bind to all receptors, so there are specialized combinational codes. And then these ligands are also different. But, and then you have the effector molecule, which transfers it from the receptor to the cell. And then the targets are the areas on the chromosome which read out the information. But here this slide shows you, you see 1, 2, 3, 4, 5 pathways. And the important point here, there are not many more. So these are the 5 essential pathways which all animals use during their development to communicate between cells. And now you can imagine, so how are these 5 pathways making these complicated bodies which you see when you just go out in the forest and see birds and insects flying around? They are all made by only these 5 signaling factors, right? So you don't need to remember them, but the key here is 5, okay? And this is in principle how you can see this information, right? So you have an address. So the signaling factor is binding to specific cells. This is like you have to write an address on an envelope. Then you have the content. So there's important information in this message, which then gets transferred to the cell, and it's a delivery, right? And this can be changed according, for example, the timing or also the concentration. So here the concentration is essential, and so different concentration can be read out by different cells and then become something different. So the delivery of this information, and now I go to the inner part of the cells, again the schematic area. So the transfer of information with a signaling factor I have shown before. So there are only 5 of these pathways, but then there are many, many more genes which are on the DNA and which can be read out to make an organism. And, oops. So, there are specific molecules in the cell which bind to the DNA, and depending on which combination of these factors, you can turn other genes on or off. And here's just a picture of these symbolized by these images, that you can have activators, so molecules which activate genes, and then you can also have repressors, so molecules when they bind to DNA, they switch off. And here you see already a glimpse of what is possible and how you can make with these 5 pathways only and a bunch of 300 transcription factors, that you can make different combinatorial codes which then give the information to other cells and then build finally an animal. And this is the way, and then of course to form shape these cells, which have to be of course some features to, for example, contract, like in our musculature, for example. When I do this lift with the arm or when I talk, muscles are contracting and relaxing so that, and these have to be signaled, and these contractions are in every cell available. And this is also important to, for example, during the development to make an animal to form an arm. Okay? So the information here is that you can contract cells in a specific region, and when you tell a cell to contract, you can make shapes like things ingress into the body, etc. So these are the essential things. And here again, the diversity of animals. And now I will give you a little bit of insight into like how do animals like develop, right? They go through specific steps. And here in the next slide, you see developmental stages, so embryos of these animals. And here are they. And they are also sorted according to the phases through which the development comes. So here, for example, the first row, the upper row, is the stage called cleavage, where the fertilized zygote starts making the first division, right? And then the next row is the phase, because then you have a ball of cells. And the goal is to form inner organs like the digestive system or the musculature. You need to put cells from outside to the inside of the animal and also to form cavities. And this phase is called gastrulation. And then after this, some animals are forming dispersal stages, like larvae, for example. And we know the caterpillar from, for example, from butterflies. They are machines for getting nutrients in to then make this beautiful butterfly or less beautiful butterfly. And I will not talk much about larvae. I've decided not to make it too complicated today. So then I will go and also talk about organogenesis. So because then when you have inside cells and outside cells, then these cells need to know what to do in which area of the body and how to do this shape, okay? So, but what was not on this picture is the fertilization, okay? This has to happen before. This is kind of the activation of the egg, right? I hope the word is correct. So to remind you what fertilization is that you have a bunch of sperm and only one sperm has to enter and activate the oocyte. And if you have many sperms entering, then it will not work because there's too much information in the cell and the cell doesn't know how to deal with additional information. So the fertilization is an important step in development. It's a starting point. And I will not talk much about it. I just want to show you that our picture, what we see in movies, etc., from sperm, they don't always look like human sperm, right? So here on the left side, we have different morphologies from different animals, and this shows you a diversity of how a sperm can look like. So it can look like a Christmas tree, it can be like only single cells which without this long tail, like crawling sperm, like they don't even have this tail. For example, in nematodes you have sperm, which is a cell which is just crawling around, okay? So, and then, but all of them, they hit at some point the oocyte and then transfer the information which is in this super small cell to the big oocyte. And this is here important. You see the relation of the size, you have one sperm only hitting a huge cell. Okay? And what I also want to highlight now is that there is the mother which produces the eggs, gives already information to the oocyte, which is essential for making an animal. And this process is called oogenesis, so the egg making, okay? And here we have a fruit fly which is studied extremely well, a model animal, and many people, over 3,000 laboratories on the world work on this animal. And therefore we know a lot about this animal and also how the fly makes the eggs. And here you see how the eggs are made. So they have, of course, ovaries, and then these eggs are built up to this end product. But here in blue, you see already that there is some information given from during this development of the egg into the egg from the mother, which determines one of the most important things, which is primary axis. So a body axis, which is important, and you see this, one top to bottom, and this is the animal vegetal axis or anterior posterior axis, and this is often in many animals the first identification and coordinate system which is put by the mother into the cell. And here you see this blue stuff, which is information. And this is the whole process where you have first some cell divisions, and then you have nurse cells, so cells which are only there to produce yolk, like egg yolk, for example, and putting this information into the egg. And then you have on the opposite side molecules, here for example the so-called EGF pathway, which is putting some information for making the axis and telling the axis, the egg, the axis, future axis of the body, so axis, sorry. And here are the molecules which are playing the key role here in the fly to determine where is anterior and where is posterior, so where is the front of the fly and where is the back of the fly. So where to put later the head and where to put the tail and the wings. So now I show you a movie of the development of a very important animal, which is a nematode, Caenorhabditis elegans. And we can see that morphology, oops, this is wrong. So this is the wrong word. It should be genome, right? The genetic information is transferred into the morphology via the development. And this is a development of this worm. So you see how the cells divide. And then there are already cells immigrated into the egg, and then you form slowly the shape of the worm, and then it hatches out of the egg. And this is a nematode which you have. And this is how the cells divide and give rise to new cells. And this is called the cell lineage of the worm. So there was a person, John Salston, who was sitting in front of the microscope and looking at each cell division of this worm. He didn't have a video recording. He was looking, taking another embryo, looking, and wrote down in notes how this cell is now dividing into this cell. And then this neighbor cell is doing this cell division in this direction and forms this cell. And he built up this whole tree, which is the cell lineage of the worm. And for this kind of work, you can get the Nobel Prize, which he also got, right? John Salston got the Nobel Prize for this, and studying how the cell tree works and how it works out to form a worm. What we don't have, of course, during this whole process of the development, again, the cells need to know what they have to do at each time of the development. So the information has to be transferred at each phase of the development to each cell in this worm. Okay? And this is happening with these molecules, and now I show you a different movie of another worm, and this is a cleavage stage, and then you have also here the shape formation, and this will become a gastrotrich, which is another worm, and then it hatches out of the egg. And then you have, of course, the information giving to the animal, and then when you take this embryo apart, you see already regionalizations. For example, there's this ring of cells, so each sphere indicates a cell, which will form the nervous system. Then the inner part here is the digestive system, the gut. So you can analyze this development, and now with video recording we have a much better information and possibilities of studying the development of animals. And there's also the microscopy, which we can use nowadays, is amazingly progressing, and this is coming with better computational power as well, because we have to store all this data at some point. And here is an example again from the fly, in which phases it is subdivided, how this information of the cells is transferred. So we see these colorful embryos, and the color of these photos is showing the expression of a specific gene, a transcription factor, which is informing the cell of what to become. And here you see that first there is the information which is put from the mother into the egg, where you form the back part of the animal and the front part, and this is illustrated with a gradient of red and blue. And then this body is subdivided into additional regions, so that the cells know I'm not here in the front part of the embryo, I'm sitting in the middle part, because also in your body each cell has to know exactly where it is. And this is based on the specific imprinting from signals in your body. And then you have this complicated thing, and here also a Nobel Prize was added to this. This is from Christiane Nüsslein-Vollhardt. She looked at mutants from this fly, and then finally also figured out all these gene cascades in the fly embryo. And this is how beautiful these development can look like when you label and stain some genes, and this is the pattern of stripes which emerges, and this is already advanced technology. But in the next I show you a video of the development of the fruit fly, which is done with the best microscopes you can have on this planet, which is, he didn't get yet a Nobel Prize, maybe he will never. It's Philip Keller from Janelia Farm, and he did the movie which you will see now. And this is the fly embryo, which has already about 500, 800 cells. And now you see the morphogenesis, and you see how these cells have to organize themselves to make a specific pattern and form the fly. Here the nervous system will be formed in the middle stripe, and you can investigate these embryos from different angles at the same time. And this is a so-called light sheet microscope, which where you can trace each cell in the embryo throughout the whole development. So what John Seilston did by hand on the microscope, nowadays you can do with a microscope, and you record everything, and then later you can analyze this, what's happening. And here you see how this will, and then of course when the muscles are formed, you will see that the embryo is contracting, and then you see that the recording is then more difficult to do. But this is how a fly is made, and then you have first the larval stage, which can crawl around, and these are the first contractions of the embryo. When it's clear, musculature has been differentiated already, and then you have these contractions. And then it goes mad. So the first cell divisions are called cleavage. And this is when one cell stage then subdivides into the next stages. So it's a simple process of cell divisions. And here you see a video which is already a 4-cell stage here. And this is how it works. So you have the subdivisions, and then the new cells are born, and at this time already the cells get the information. Each cell at this time knows already what about they will become. At least they know where if they will be in the anterior part, or in the frontal part, or in the back part of the animal which is born out of these embryos, which I will show you soon what it becomes. And you have here already the internal cells, and it continues that you have cell divisions after cell divisions, and you also see the protection shell around this embryo, where you have like, so that nothing bad happens to this. And this is what develops and hatches out of it. It is a larval stage, which is a dispersal stage, which is extending now, and then it swims away. And this larva will swim around and then at the end become like a bryozoan animal, which maybe you have seen them on shells, sometimes when you eat mussels. You have on these mussels these animals sitting. They are of course dead when you boil the mussels, but these are beautiful animals, and this is just to show you what the development can do. But in cleavage stages, there's another important process happening, because I told you already that there is anterior, posterior, or front and back part information already put by the mother into the egg. But then you need another axis development. So for example, the cells in the body have to know if they are more on the back part or the frontal part. And this information is usually given during the cleavage process to the embryo, and that, okay, you are you are along this axis, but then to make a bilateral symmetric animal, which is an animal which has a left and right axis, and a back and a belly, for this you need additional information. And in this bryozoan, this is happening by this single cell, which is labeled in bright, which is making a specific pathway, the MAPK pathway, and it's expressing the pathway and telling the surrounding cells, hey, I'm the most back cell in the future embryo. And so you know that you are not in the back, you have to do other things, okay? And this is the information done by a single cell, telling the other cells, hey, you should not become what I am, okay? Then I told you the next step is the time point where you have this ball of cells and you put cells inside the embryo to form the digestive system, for example, also the musculature. So all your inner organs have to were built by cells which integrate during embryogenesis into your body, okay? And this process is called gastrulation, right? One famous embryologist has said this is not birth, not marriage, not death the most important thing in time, it's gastrulation. Obviously a nerd, Louis Wolpert, but you see that you have here some embryos gastrulate with only 2 cells, which then found the whole inner organs, and some embryos gastrulate with a whole sheet of cells, and there are already a lot of differences between these embryos, what we find. And this is what happens. So this is like kind of the information the cells receive during the gastrulation process. So here in these different colors are molecular information which is given to the cells, which then regionalizes already the digestive system. So here you see these cells integrate into the body, and then the cells know already, okay, here the bright blue, I'm on the tip of the gut, right? And I will later connect to form the mouth in the embryo. And here you also see what is important, you have the first shape changes. So to put cells from outside inside, you have to contract at specific parts the cells on one end, and then you bend in the tissue, and then you have the immigration also from other cells in this process. So constriction here plays an important role, and to make shape, the cells have to have these abilities to really make shapes and contract at specific parts. So this is the process of gastrulation. And when I show you these colors, right, these are 4 colors, right, and then, okay, this is fine, but in the background of all this information during this process, I just show you a complicated picture, which is this one, and this is an interconnection of genes which are active during the process. So in the sea urchin, which just forms a spiny bulb of an animal, which has nice gonads, which are tasty, you have all this happening in this information cascade. And you see this is very complicated. I, of course, don't go into details, but this is what's going on in this cell. And this is, of course, also allowing variability, right? You can imagine that when you change something here in the pathway, also in evolution, you can make new animals, because there's a lot of information happening during gastrulation. And here you see a gastrulation of a frog. So when cells immigrate, and now pay attention to this movie, what shape changes they are happening. So what you can imagine, what information has to go through the cells to make these contractions and make this shape. And this was the gastrulation, and the next step is the neurulation. So because you know that the nervous system is also inside in our body, and this also needs to be transferred. And this is happening in the second phase here. So this is the gastrulation. This will form the digestive system and musculature. And now on the outside, this will be the nervous system, the backbone of the frog, right? And this also needs to integrate into the animal. And this is a lot of complicated changes. And then it's of course a question, so do these, I told you already that these animals are using 5 pathways only. And these are, in principle, the communication, the signaling wires, right? Like you have a phone line and you have an internet connection, so these are 2. They have 5, and then you need other factors. But this is showing you, these are specific transcription factors, which are the HOX genes. And here is such a HOX transcription factor symbolized with these spirals, which binds to DNA and switches on genes. And this slide shows you that all animals use the same genes also for making, for example, the regionalization along the body. So you see the fruit fly, and this is what happened in these developmental biologists who are specialized on studying these genes. They meet on conference, they are fly researchers, and then they meet mouse researchers who look at mouse development. And then they, hey, you are talking about the same genes. This is the same gene what I have, right, in my mouse. And then they communicated, and then they discovered, wow, this is amazing. So the same genes are making the front to end patterning of a fly, as well, which are expressed from front to the end in our backbone. So the identity, so that the vertebrates which make ribs, for example, are not here on the further back, this information is given by a combinational code of these HOX genes, which bind to DNA in a specific pattern. And then the cells know I'm not in the brain, I'm not in the belly, I'm in the back, in the vertebrae. I will make bones. Okay, and this is what the information is happening and transferred to the cells in this process. And now I show you another amazing movie from also Philip Keller again from Janelia Farm, who studies also zebrafish development. And this is one of the most advanced microscopes. And this video just has been published like 2 months ago. And this you will see, you see a ton of cells, all these blue dots are cells, and then you have a specific label of some cells which will give rise to the nervous system. So this means you can track each cell in this embryo, and then you can also identify using genetic labeling which cells is becoming what and where are they at which time point of the development. So this is the outcome, but I will talk here mainly about I will show you how the nervous system is developing. So again, gastrulation, and now it's the nervous system, so they come together, these cells which are orange, and they of course do cell divisions and then will form here the nervous system. And this nervous system will integrate into the animal embryo, and that's it. So you can also see this from different angles, from the side, etc. And this is the shape changes. And this is just to remind you that there's a lot of shape changes necessary to form also a human. So this is a kidney, and you see that these kidneys are made by many, many channels, and each cell needs to know which position they are in. And here you see the development of our kidney. And then you have here specific regionalization. Each cell needs to know what they are doing. And these are the genes which are telling each cell and which correspond with between the cells to make a specific outcome, namely a very complicated organ, our kidney, with all these channels and canals. And this, of course, has to be functional. Okay. And not only cell divisions, but also programmed cell death has a specific role. For example, our hands, when they develop, they start kind of as a bulb, which looks like a fist. But then, during the development, you have an outgrowth. And then, between the fingers here, you have programmed cell death. So there, suddenly other cells tell these cells, you have to die. And then you get a hand, right, out of this. And here, for example, you see how this develops. And here you see the area, here in orange, the cells, which are told, you have to die now. And then the material gets engulfed, and then you have a specific pattern. And this is also how shapes are made, namely our hand. And this is the same in horses, etc. So how this happens. And now I will come to my last slide of this presentation. And I hope I told you already that it's a very complicated process which has to be organized in a well order, etc. And many things can go wrong. There are buffer areas where you have a control, where something, this doesn't work really well, but then we have another pathway which jumps in. But sometimes, of course, now it's also the most ugly picture, but it's a nice story. So you have the cases where things go wrong. And this is, of course, everybody knows this. You can have malformations during the development, which can affect many organ systems. And also another thing is that cancer, for example, is an outcome of miscommunication and misidentification of cells in your body. So when you have, for example, a cell in your body getting the wrong information by mutation or something like this, you can have cancer as a disease, right? And this is what's happening. And the same pathways which I've shown you before are also involved in cancer as a disease, right? Then signaling pathways are expressed too much signaling is going on, and then you have these cells which go crazy, right, in your body. And this is not what we want. And this is an interesting case which combines kind of these malformations with also cancer. And this happens so in the US, in Idaho, there was a meadow, and the calves from the cows were eating there, and then with a high frequency, there were calves born which only have a single eye, right, here. And this is this ugly picture on the slide. So there's a calf, a young calf with only a single eye. And what they figured out is that on this meadow, there's this plant growing, right? And this plant has a side product, which is this molecule. And this molecule is interfering with an important developmental pathway, namely the hedgehog pathway. It stops it. It breaks up the communication of this pathway during the development, and then you have not a bilateral formation of two eyes, which, yeah, you all have this. And so this miscommunication leads to the point that you have only a single eye exactly in the middle. And this is, of course, reminding us of Cyclops, right? The Greek fairy tale organisms or human-like organisms which only have a single eye, right? And this is why also this product, which was not known before, and this is of course based on science that the people figured out, it's this molecule from this plant. And when the calves are eating this, then they were born with only one eye, because the hedgehog pathway is very important in this stage. And they called this cyclopamine, right, from Cyclops. And investigation and science went further, because now this molecule produced by the plant is used in cancer therapy. Because some cancers are based on an overexpression of hedgehog pathway. So you can identify cancer, okay, here, this is wrong, and then this molecule, slightly modified, is used for chemotherapy of these cancers, right? So this is a whole story which kind of connects these two, the malformation which was observed in a calf, then up to cancer therapy, to just show you there are important pathways which play a role, and when something goes wrong, then you need to have a good therapy for doing this. And so all this is, of course, yeah, this, as I said, my last slide, and my research group is here in Bergen, and this is our photo with some people disappeared already and went up to form their own research groups, and then the funding comes from here, and of course, again, thank you for also this nice location. I've never been here in this beautiful building. And I want to thank you also for your attention, and want to finish with a nice last picture. Thank you very much. So I think you can ask me questions.
Давайте вопросы. И за лучшие вопросы у нас есть 7 книг от траектории, посвященные социальному поведению животных.
What is the main liquid in first cell?
Yeah, so the question was, what is the main liquid in the first cell? So there we have a lot of nutrients, but it's also packed with lots of protein, and okay, nutrients are protein. There is also a lot of fat, right? Because fat is energy, and there's a lot of molecules which are lipids, which then provide the energy for the further cell division, because all cell divisions cost energy, and then you have it stuffed there. But the molecules which the mother puts into the egg is messenger RNA, which the mother produces and also puts in a specific region, right? The egg is looking like a round ball, but it's already subdivided into regions, which then get read out by the different cells. But it's mainly storage. It's like when you have a chicken egg for breakfast and you open it, and all the stuff inside is very nutrient and protein rich, and this is what is in the egg.
I would like to ask a question about genetic modification. Lately, as I read, in China there were 2 pigs born with the cells of monkey for grown-ups human beings. And I want to know, when people use genetic modification, do they attempt to pay more attention to DNA and DNA replication, or to hormones? Which of these 2 things play more bigger role in creating different patterns?
Yeah. So usually what the people do is going for the DNA, so for the chromosomes, and do modification there. Because one reason is because the DNA is also the molecule which is transferred to the next generation, right? So when you have the sperm swimming, it carries DNA, and the egg also has DNA, and they fuse together, and this is why we have elements from our parents, from both parents. And when you do a modification there, then it's transferred to the next generation, right? And this is why they focus mainly on this and not so much hormones or something like this. Yeah.
The smallest listener.
Ah,
He's very nervous. Чуть попозже, ладно, нет, ты сам меня заговорил.
So I do have a question. You showed the images when there was the baby fruit fly, yeah? And so the cells were moving, like kind of 100 cells. They were moving inside, choosing a special place to form like the nervous system or another system. So what does actually help them move to one special place? Is it electronegativities like potassium and sodium ions, or is it something different?
So for moving of cells, if they are not pressured by the external contractions, then when they move, then they have these like extensions from the membrane, which are philopodia, right? So these extensions make them attach to different size. So in principle, these cells can crawl along an area where they have information. So also there the information is giving, okay, this is the road you have to migrate. And then these cells can go into this direction.
So I have also another question about the type of cells. So as you showed us, at a certain point there were a construction of muscles. What needs well, actually, some nervous cells to be created.
Yes.
So, at the very first cell, there's like only one unique type.
Yes.
During the division of cells, how one certain type of cells formed and not another?
Yeah. So the first cells in an embryo you can see as a stem cell, right? So it is multipotent, it can become everything. But then there's already additional information for each step, and the last information a cell received is the information of what cell type do you become, right? So there is a specific molecules expressed in each neuron, for example, which is ELAV, which is a molecule which then binds also to DNA and makes a specific neuron, right? And then the cell knows, okay, I will become a neuron in the future. And then these cells get the information to connect specifically to muscles. Because at that time, the muscles also know already, okay, I will become a muscle. And then they get the information from the nervous system and neurons to contract. And then you have, it was a good observation from you, of course, you need a nervous system to have these contractions, and at the same time, also the nervous system develops to make these contractions.
Maxim, thank you for the lecture. It was very interesting and I think comprehensive. So in this year, a very interesting work on squids, when scientists measure the expressions of certain genes which are involved in the formation of appendages of vertebrates and arthropods, and to some extent of annelids. And they made certain hypotheses that it is unlikely that our common ancestor also possessed appendages. And they made deep knowledge that there exists some conservative gene regulatory network which regulates the formation of appendages, syncytia or something else, which were very primitive in our last common ancestor, but developed into something more complex in different bilateral lineages. So the question is, do you believe in how can you comment this concept of deep homology? Is it likely that our common ancestor possessed some primitive appendages like antenna of Anomalocaris or something like this, or Picaya?
Yeah. Okay. Yeah, thank you for the question. It's a specialist question. The reference here is to a gene cascade, like a combination of transcription factors which are expressed in the same arrangement in the leg of a fly and also in the octopus arm. And the idea here is, so is this indicating, when it's the same transcription factor cascade, that also the last common ancestor had legs or something like this? And this is usually called deep homology, which means homology is a common origin, and deep in time means very, very long time ago. And so what this study did not do is that they looked for these genes where they are expressed in animals which don't have legs, right? Because what we know that these genes are present in animals which also don't have legs, right? So they haven't lost these genes, they are still present. And which is fun, because my lab is now looking for exactly these genes in animals which have no legs, right? Because we are looking at this, and we found, for example, that these genes cascade is also expressed in a small extension of a larva from random worms, phoronids, right? And you have this expression in everything what is outbudding kind of the body. And one of the tip genes, distalis, which is expressed on the most distal part, on the tip of each arm, which is, for example, also expressed in nervous system, right? And the idea connection here is that for making antennae and sensory organs, it is of advantage to have an extension out of the body. And that this gene cascade was mainly there for sensing the outer world. And in some animals, this out pockets were to evolve to widen the range of feelings. Because also at the tip of the octopus arm are neurons, right? And they also need to touch and catch prey. And this is likely the primary function of this cascade is in the nervous system, and then later got used multiple times, maybe independently, for making also outgrowths out of the body. And this is a common thing in what we observe when we study different animal development, that the same tools have been used over and over again. And I use deep homology only for things we don't know yet, because the solution for this question is not yet known, and we will know later, and then maybe we don't call it deep homology anymore when we have really an idea how this evolved.
Thank you so much for the lecture. What I wanted to ask is about the transfer from one-cell organisms to multi-cell organisms. And you mentioned it is very important how the cells communicate between each other. And the question is this process different in the other types of living beings, like in plants or fungi, than in animals? Is there something special about cell communication in animals?
Okay. Yes, so what is known about, coming from a single cell organism to a multicellular organism, There is one pathway in the animals which this happened, but then there's another way. Of course, plants have done this also, and fungi, so when you think about eating mushrooms, they also have become multicellular, right? And these events happened independently in evolution, and interestingly, they use different pathways. So the animal-specific pathways are specific to animals. Like these pathways I've shown you, they are not in plants, right? So these are specific animals for making multicellular animals. And this is what has been used, which was present in these like single-celled organisms. Many of these genes were present and then leading to multicellular animals. But the animals use different pathways than plants and mushrooms.
У меня два вопроса.
I have two questions.
Первый вопрос. Это почему у некоторых людей бывают одинаковые уши? И что за это отличает какой-то ген или клетка, которая отличается от
First question is about ears. Why different people who are not relatively connected have the same ears in the cell? Or is it a gene that is responsible for this kind of stuff? What's the second question?
Yeah. So the question was why do people have different ears, right? But same ears. So
They're not connected. They are not relatives because he.
У моего преподавателя в театральной студии с какой-то девочкой тоже из театральной студии одинаковые.
His theater teacher and a girl who goes there together with him, they have the same ears, same form of ears.
yeah.
Important to know why.
Yeah. So you have to look very carefully at the ears. They are not always the same, right? So for example, here this can be this can be large, and some people don't even have this, right? And this is genetic, yes. This is molecular information which often comes from your parents, which makes the ear shape. But it doesn't mean that when your friend has the same ears as you, that the parents are the same.
Почему, когда образуется рука, почему сначала вот так вот образуется, почему зарядка клетки делится? Лучше бы сразу вот так делиться.
Okay, second question is about forming of our palm. When it's in the stage of hand, like in the embryo, why is it necessary to become this instead of this at once? It's a waste of time and cells.
Okay, yes. That's a good question. I might not have a good answer, but it could be that it's an evolution question, because vertebrates, this would be now my answer, there are vertebrates like lizards or birds which have not 5 digits, but only 4, right? So when you start from one, all start with the same starting point. Also birds, when they make the wing, right? They all start with the same point, but then doing the programmed cell death can decide to have only 4 fingers or a bird wing. And this is the ancestry, I would say, which imprints this similarity at the beginning and then the differences. And for this, it's waste of evolutionary time, maybe, but it's easier to make differences by starting this way. I hope this is a satisfying answer.
I should remind you that we should to fix the best questions for the book.
Yeah.
Once again, thank you very much. This is interesting. For me, I really like the picture presentation with a cell tree. One cell makes different types of cells. If we think that we want to change, let's say, some organ in the body which functions wrong. So, like, at what stage do we take? Does it go the very same process as it goes in the whole body? You know, like from multiplying and diversifying the cells, or is it a different process for making? Like, is it a different process for making like single body part and for making the whole body? I mean, like, does it go with the very same process of forming the organ? Different process?
No. The sequence of body part formation is different between species. So, or let's say larger distances. You can make first the nervous system and then the musculature. You can also first make the musculature and then the nervous system, for example. So there's a time shifting possible, right? But of course, the cells have to be internalized to form the inner organs, and this is the thing. But between species, there's a different organization of when you do what. And with larger evolutionary distance, the differences can be very, very different. So, and this is called there is a name for it, heterochrony, which means a shift of timing of developmental processes to each other. And heterochrony from the timing, different timing. And this is not always the same.
And for example, we have an animal who has probably liver and went to make another liver. Will the process of kind of building up from a single cell to the whole liver be different like for the embryo and for the liver under the microscope or like wherever it's
So the adult processes to make a liver is very similar to making the embryonic liver. So there are a lot of similarities, and this is also why cancer is using the embryological pathways for forming tumors. So it is all these developmental processes are still active to some extent in our body, and this is why our cells have the information. But during the embryonic development, there is a possibility that neighbor cells can replace the function of a cell which might die, right? It can happen in the development of humans that in the very early stages suddenly a cell dies which was supposed to give rise to the liver. And then the neighbor cells says, oh shit, yeah, it died, so now my function is to replace the cell, which is more important than my function to do the other stuff. So there is this rescue point to some extent in embryonic development as well.
Thank you for the lecture. It was very interesting. My question is, you said that one cell generates molecules that then transfer to another cell, and this changes the weight to left. My question is, how do molecules know which they are frozen.
Okay. Yeah.
The first cell that generates or unable to spread around, or it has some mechanisms that said that this one goes in that direction.
Both is possible. So it has been observed in animals that there's one plane diffusion, so it's more random, right? So that you can, one cell secretes ligands throughout and they spread equally to the neighbors. And then it's depending on the concentration. So the cells very far away receive only one or two of these molecules. And the closer cells, they receive more molecules, and above a specific threshold, then this cell becomes the information to become a different cell. But also in the, for example, making the fly segments, and likely also here the caterpillar, the segmentation, the indentation, there is a guided information. So the information, these ligands are transported only in one direction and not in the other. And this is even better for making sharp boundaries and giving not the same information to the other segment to really make sure there is a straight line, right? And also the stripes in Drosophila, where you have these nice stripes in the embryo, these are also often made by directed information. So it's both happening.
Could you please recommend some literature on the topic of cell communication and multicellular animals formation?
So I started with by myself reading Scott Gilbert's book, Developmental Biology, and there you have the whole information in this book, from the molecular pathways and also evolutionary aspects there. So.
Could I ask about forces in the process of development in the cell? You told about 5 mechanisms of communication between cells, but if the forces like electromagnetic or magnetic fields or maybe gravity influence on the process of developing cells, maybe if we have a strong magnetic field, we have a big caterpillar in one direction.
Extended. This is an area which is not well investigated yet. So the impact of gravity on embryogenesis is, of course, the best experiments are done in space, right? Because then you can exclude gravity. Otherwise, you can, of course, put them in a centrifuge and then study their development afterwards, so where you impose different gravity. In many cases, it does not really play an important role, but there are some described places where it matters, right, the gravity aspect. And also, for example, that you have ion gradients, like electric gradients. This, for example, in the chicken embryo is an important thing, that you have a specific gradient of electricity there to then make and organize the cells and give an information. And so there are some rare cases, yes, but mainly it's going by these molecules.
Thank you very much for the lecture. I have a question about prolonged death of cells. I think it's not a secret that we die at this or that period of time, like we have length of life, and it depends on the food we eat, on the place where we live, right, and stuff like that. Do we have, from the embryo stage, do we have pre-programmed death cells or something like that that influence length of life?
That's going a little bit out of my area. I'm not sure about the status, what is now. There was a time when people said that the telomeres in the chromosomes are important. So, for example, you have each of these DNA bases, they have a tip of which is not copied or only partially copied to the next generation of the cells. And then when the cells have done sufficient or a lot of cell divisions, then they know, okay, I'm over, I don't divide anymore, right? And this is in fact happening in our body, right? So at some point the organs do not get replaced with fresh cells. There are no cell divisions anymore. And then, so it happens usually, what I've heard, every 7 years, where our whole body, or nearly the whole body, is replaced by new cells. And then after a number of times, it might be these telomeres, but it might be something different now, what the people look at. But there is a kind of a program there. There is an element. And this explains why a sea turtle can become 300 years or 400 years old, and then humans, they have their limit around 90 or 100.
Thank you for the lecture. I had a question about the way modern scientists can make tissues in cell cultures from undifferentiated cells. As far as I know, it is possible, and is it possible nowadays to build organs like, let's say, liver or a part from undifferentiated cells? And how can it be done by a genetical regulation or by adding some differentiation factors into cell culture?
Yeah. This is now totally in fashion to do this, right? Building new organs out of cell cultures. And with a goal, of course, in medicine to make at some point new hearts just by taking like stem cells and giving them the right information. And this is here what is important, right? You need to add them the correct information that they know that they should become a liver cell. Right? And then they build a liver. And some cells are really organizing themselves into specific regions, even autonomous, so you don't have to add something as a liquid or a fluid to this, which gives them the information. And but this is the main goal here, to get organs out of the dish, which then in the optimal case get implanted to the human body to replace sick organs. Yes.
А есть ли существо, которое вечно живет, например, медуза или лобстер?
Are there any immortal species like jellyfish and lobsters?
Okay. In principle, yes. So jellyfish is a good example. So you have there like hydra, which is a well-investigated relative of jellyfish or so. They make new hydras all their life, and you cannot even say when they are dead. Of course, when you smash with a hammer on the animal, then they are also dead. But they don't die by themselves. So they can reproduce new and new offspring, and there's fresh cells everywhere. And this is also why people study them, to maybe find the secret of aging there, and that one can stop the aging.
So the body they had, it dies, and they produce the new one?
No, the body stays alive.
But cells, some cells die and they produce...
Yes, yes.
To continue the question with I asked, for example, if we have a human being or an animal has failed to liver, how do we make another one? Do we take a cell from that human or that animal, fix it and make another liver, or we take somewhere some perfect one, or somehow build it up and then we form it a tissue and an organ and we then implant it so that it doesn't have problems with doesn't help, has a problem with the body resistance and accepting that.
Yeah. The ultimate goal and what people try is to use human stem cells, so undifferentiated cells, and from the same human if possible, right? And then you can extract these cells, give them the information to build a liver, and then put the liver inside. But of course, the science starts first with trying to do this with pigs, for example, like using pig cells, but then we know that there's a huge resistance, or can be a huge resistance of the body to expel the organ, right? But people managed already to build, I mean, when you use stem cells of an animal, you first start with the pigs, for example, and try to establish the methods there before you do this with human stem cells, right? So this is science in progress.
So as you just mentioned, during our early growing up, we retrace the path of our ancient ancestors, so that's why we look like a fish and frogs. And so, is it somehow possible to speed up, to accelerate our growing up, at least in these the first months at least, and not to pass that other stages, and to come directly to the shape we want?
This is now a guess also, a guess answer, because I don't know if somebody wanted to speed up the development of any animal or so. But what is here the case that the timing is very important. That each species has a specific timing of when to develop what, and the whole information transformation is orchestrated according to this timing, because timing is here important. Also, like when you think about the diffusion of a molecule to the next, it needs to diffuse and needs a specific time for this. And then when you speed this up, everything would have to happen faster. And there are chemical processes, there are physical processes, and all these processes have different speeds. And this is, for example, you cannot really speed up a development of an invertebrate. You can speed it up a little bit. I know, for example, when you record the development of a worm, like Cenoaptetes elegans, you can do this at 24 degrees or 31 degrees. And they use a different timing for this, but you cannot go to an extreme there, right? It still needs to function, and the first process, which then doesn't work, stops the whole embryogenesis.
Thank you.
She wants a book.
Yes. We have already talked about one of Greek ancient cycles, like mythological creature, and I was thinking about another one. Might it be possible to form a totally new organ for example, for a human being of another incompatible creature like a butterfly, to make wings, a butterfly wings for a human? Is it somehow possible?
This is a good question, right? It's about a specific, what is often used as a constraint in development. So can it be that we are born like angels, right? Angels have arms and legs, but then they also have additional wings, which look very much like bird wings on the back. And so Louis Wolpert, he used this slide of an angel in a developmental biology talk, so this is the guy with the gastrulation is the most important thing in your life, to exactly raise this question, right? Can we ever be angels in a way that we have wings on the back? He said no, right? Because the material is not really there to make wings. And I wouldn't say totally no. Not in our life, yes, but who knows where human would be evolving, right? We have other cases where there are animals having evolved a new wing, additional things, and why not? You just need to give it a specific time there. But it will not be a butterfly wing likely. It will be more likely like a bird wing or something like this.